faithfully delivering value by interpreting great books from around the world. Hello, this is Sally here with Book Talks. Today, I'll be interpreting for you the book, The Double Helix, the story of the discovery of the structure of DNA. On April 25th, 1953, the top academic journal in biological sciences, Nature, published a paper titled Molecular Structure of Nucleic Acids. The paper proposed that deoxyribonucleic acid, commonly known as DNA, is a double helix structured genetic material. This discovery sparked a revolution in the field of biology. It laid a solid foundation for understanding cellular structures, functions, and operational patterns within living organisms, as well as human genetic development and aging. Beyond that, building on the discovery of DNA structure, humans invented various advanced biotechnologies such as hybrid rice and genetically engineered recombinant vaccines, which have had a huge impact on human production and life. This paper, a mere 128 lines long, is considered the most significant achievement in 20th century biology. Its authors, the American James Watson and his British colleague Francis Crick, jointly received the Nobel Prize in Physiology in 1962. The author of this book is James Watson. He was born in 1928 and was only 25 years old when he discovered the double helix structure of DNA. So, what enabled this young man, just out of school, to discover the greatest treasure in 20th century scientific history? And what are the little-known stories in the process of discovering this treasure? Today, through Watson's memoir, we'll journey back to that magical treasure hunt together. Major scientific discoveries are often the result of countless scientists working together in succession. The double helix structure of DNA is no exception. In 1865, the Austrian scientist Mendel discovered that organisms of the same species share striking similarities, and these traits are passed from parents to offspring. Thus, he first proposed the concept of hereditary factors. In 1909, the Danish botanist Johansen replaced the term hereditary factors with gene. However, until the 1940s, scientists still hadn't figured out exactly what a gene is. To solve this mystery, many scientists joined this treasure hunt one after another, and our protagonist Watson was among them. So how did Watson embark on the journey to discover the structure of DNA? Interestingly, James Watson, who would later achieve dazzling success in biochemistry, had no interest in chemistry when he was young. During his undergraduate studies at the University of Chicago, his dream was to focus on studying birds. He hoped to become a museum curator someday. Watson began developing an interest in genes. This was because he read Schrödinger's book.
Many people know this famous physicist because of his cat.
But actually, Schrödinger himself was a pioneer in molecular biology. In his book, What is Life?, he wrote that genes are key components of living cells, and to understand life, we must understand how genes function. Watson's interest in genes was completely ignited, so he decided to study genetics. He first applied to Caltech, where a Nobel Prize-winning chemistry heavyweight who had deciphered protein helical structures was based. Linus Pauling. Please remember this name.
He'll reappear repeatedly as Watson's main competitor later in the story. Pauling rejected this young man who knew almost nothing about chemistry for his PhD application. Eventually, Watson went to Indiana University.
However, at this point, he still hoped to solve the gene problem without learning any chemistry. His supervisor initially encouraged him to learn organic chemistry, but after Watson directly heated benzene with a Bunsen burner, after causing an accident, the supervisor decided to just let him be. After all, letting a PhD student muddle through to graduation seemed better than blowing up the entire lab. After earning his PhD, Watson went to Copenhagen, Denmark for postdoctoral studies with a fellowship, continuing his gene research. His research focused on bacteriophages, viruses that specifically infect bacteria. Because of their simple structure, studying gene replication through bacteriophages was an effective approach. During this period, Watson conducted many experiments and published some high-quality papers. However, he didn't undertake reproduction research because that required understanding DNA's chemical structure. And now, unless Watson became a biochemist, he didn't know how to proceed with that work. In the spring of 1951, tired of Northern Europe's cold weather, Watson decided to go to Naples, Italy to quietly study genetics literature. Just before departure, he got his alma mater, Indiana University, to cover his travel expenses under the pretext of attending a local conference on macromolecular structures so he could enjoy Naples' warm sunshine. Watson's anxiety about accomplishing nothing grew heavier. Due to his lack of chemical knowledge and disinterest, unraveling the mystery of genes still seemed like a pipe dream to him.
However, at this academic conference he'd only used as a travel pretext, one person's speech gave him new inspiration.
This person was Maurice Wilkins, a physicist who graduated from the University of Cambridge in England. During World War II, he participated in the Manhattan Project, America's atomic bomb development program. After the war, he researched genes at King's College London, specializing in X-ray diffraction technology, essentially using an X-ray camera to photograph DNA in cells. He then analyzed DNA structure from the developed photos, similar to how we get chest X-rays when sick. Because DNA is incredibly tiny, only special instruments and advanced techniques could produce relatively clear images. Wilkins was a master in this field. During his conference presentation, he showed a DNA X-ray diffraction photo that instantly captivated Watson. If they could photograph DNA, surely this method could determine its structure. He decided he must find a way to work with Wilkins. The next day, an opportunity came when all attendees were invited to visit a nearby Greek temple. Before departure, Watson seized a chance to approach Wilkins, who showed little interest in this young man stammering about his DNA fascination. Instead, Wilkins chatted happily with Watson's attractive cousin, who accompanied him.
Watson felt fortune had smiled on him.
If Wilkins fell for his cousin, gaining access to Wilkins' lab would be much easier. Unfortunately, Wilkins vanished alone after the tour ended. Dejected, Watson tried to forget Wilkins but couldn't shake that DNA image from his mind. So he searched everywhere for an opportunity to learn X-ray diffraction techniques. Coincidentally, Professor Perutz at the Cavendish Laboratory, Cambridge University's physics lab, needed assistance. So Watson packed his bags and left Denmark, heading straight for England. Professor Perutz had originally hoped Watson would assist his student Kendrew with hemoglobin research at Cambridge. However, Watson quickly hit it off with another colleague in the lab, Francis Crick, who would later share the Nobel Prize with him. Crick was already 35 at the time. Having dropped out during WAO to help the British Royal Navy develop mines, he was still an obscure PhD student who occasionally did experiments but spent more time pondering theoretical issues like protein structure.
He often got wildly excited about new ideas and told everyone, and with his booming voice, he always energized the lab atmosphere.
Of course, Crick was also mercilessly loud in criticizing dissenters, so he wasn't particularly well liked. Many viewed Crick as an arrogant, boastful playboy. Beyond that, Crick had keen observational skills, digested information rapidly, and could quickly design experiments to prove his theories. This intimidated many colleagues. In Cambridge's cautious academic tradition, Crick's style was truly unconventional, but Watson disagreed. To the newcomer, Crick was among the few at Cavendish who believed DNA mattered more than proteins in genetics. Plus, with Crick's physics background, Watson avoided lengthy X-ray technique study, perfect for him. From Crick's view, though he had unique DNA structure ideas, Cavendish's main focus was protein research. Now with this American eager to discuss genes, Crick no longer had to shelve his DNA ideas. They felt they'd met too late. After just one lunch discussing DNA structure and days of collaboration, They became excellent partners. All right, now the three main contenders in the DNA structure race, Pauling from Caltech, Wilkins from King's College London, and the then unknown duo Watson and Crick, are all poised at the starting line, ready for action. Next, let's explore the little-known stories along their journey to the finish line. Pauling, across the ocean, was first off the mark. He'd proven the alpha helix structure of protein polypeptide chains.
What does that mean? Let me explain.
Since the 19th century, scientists had studied proteins for over a century, understanding complex protein macromolecules. These macromolecules consist of polypeptide chains. Later, compositional analysis revealed chains contain over 20 amino acids, but how amino acids formed polypeptides remained unknown. If we compare a protein molecule to an ornate castle, polypeptide chains are its bricks, and amino acids are the sand, gravel, and mortar within those bricks. Scientists knew how to build castles with bricks and understood brick composition, but the missing piece was turning raw materials into bricks. Pauling solved this. His solution tool was a molecular model set. Superficially like Lego bricks, this model allowed assembly based on amino acid structures. Through chemical pattern connections, he discovered the brick's core structure. Amino acids linked end to end in spring-like helical coils. This alpha helix structure later earned Pauling the 1954 Nobel Prize in Chemistry. When Watson and Crick learned this, they immediately realized the same approach could solve DNA's structure. Like proteins, DNA is a molecular castle with nucleotides as bricks. While nucleotide structures were known, their assembly pattern wasn't. They needed DNA's architectural blueprint. Watson and Crick decided to adopt Pauling's molecular model method. Back then, they thought they'd crack the code quickly. Crick was even optimistic, believing that just 1 week of tinkering with molecular models would surprise the world. However, Wilkins doused their enthusiasm. After learning of Watson's idea, Wilkins and his colleague, physical chemist Rosalind Franklin, bluntly told them their model was too presumptuous. Right. If nucleotides were connected end to end in a long chain like their model suggested, DNA would appear much smaller than in Wilkins' X-ray images. If Wilkins' camera captured a baguette, Watson's model produced a noodle. Undoubtedly, this huge discrepancy meant something was wrong with Watson's model. Why such a big difference? Wilkins told Watson his analysis showed DNA wasn't a single chain, but multiple nucleotide chains twisted together into a helix to create the image effect. Whether 2, 3, or 4 strands required more experimental data to determine. Franklin openly scoffed at Pauling's model approach, dismissing its ability to reveal DNA's true structure. She believed experimental data was the only valid foundation. Everything else was nonsense. Clearly, Wilkins' results were more credible. Watson and Crick had to stop and rethink where their building blocks went wrong. And in round 1, Pauling moved fast, but Wilkins seized the initiative. Watson and Crick seemed stuck doing futile work, falling behind. The setback devastated Crick. His DNA interest plummeted. With no experimental progress, he nearly got expelled after fierce clashes with lab director Sir Bragg over credit in papers. Crick's streak of failures continued until Watson's mentor Perutz validated Pauling's alpha helix using X-ray diffraction. This proved molecular models could perfectly match X-ray diffraction results. That means Pauling's approach was viable. Given this, Watson and Crick needed to redesign their molecular models.
Just then, Wilkins, the frontrunner, hit a snag. During his research, he captured a new type of DNA photo unlike previous ones.
He named the structure B-double helix. What he and Franklin had captured earlier was called AD-DNA. When determining which structure represented true DNA, they clashed fiercely, neither willing to concede. They finally agreed Wilkins would measure B-DNA structure. Franklin would measure A-DNA's structure. In their rivalry, Wilkins consistently failed to obtain clear enough images. Franklin got sufficiently clear photos but refused to share them with Wilkins. Though their labs were meters apart, their relationship was explosive. Later at a conference, Franklin presented new findings that sparked Crick's inspiration. On the return trip, he lost himself in calculations and developed a new DNA structural model. Nucleotides, the bricks, are made of three components: sugar, phosphate, and base. In their new model, phosphates and sugars linked to form a central backbone, bases arranged outward like layered branches. Three such chains twisted together at specific angles, spiraling upward to form DNA. This model appeared to match Wilkins and Franklin's X-ray diffraction photos perfectly. Watson and Crick immediately called Wilkins, hoping for quantitative data to verify it. Shocked, Wilkins said, he and Franklin would take the train to Cambridge the next morning. At the meeting, Crick extravagantly promoted his triple helix model, but Wilkins dismissed it as hot air. Results needed X-ray verification. Franklin thought Crick's idea wasn't worth the fuss,
especially when Crick claimed magnesium ions acted as glue binding nucleotides.
Franklin bluntly pointed out that magnesium ions in DNA are tightly surrounded by water molecules, forming a thick shell. What does that mean? If magnesium ions were glue-bonding nucleotides, water-surrounded ions would be like glue wrapped in plastic bags, completely non-sticky and useless for bonding. Therefore, it couldn't be a tightly structured central axis. Only then did Watson realize they'd made a huge error. They'd miscalculated while buried in their work. Franklin's talk revealed DNA's water content was 10 times higher than their model, rendering it useless. Crick still refused immediate surrender, trying to secure Wilkins' cooperation for the next experiment. But Wilkins wouldn't waste more time and left without looking back. Thus ended round 2 of the DNA structure race, with Wilkins and Franklin steadily advancing. Though proactive, Watson and Crick suffered a crushing defeat. They realized success required starting over completely. Continuing clashes with Wilkins meant no more data from King's College, let alone conference invitations.
Worse, their failure spread through Cavendish. Director Sir Bragg deemed it wasted time, halting their DNA research. Crick resumed protein work, while Watson switched to tobacco mosaic virus. Its RNA might provide DNA clues later.
As Watson and Crick stalled, news arrived that Pauling across the Atlantic had developed a DNA structure.
Pauling's son Peter was then studying at Cambridge. Watson learned this from Pauling's letter to Peter, but without details. Pauling's model remained a mystery. Watson also told Wilkins this news. He'd hoped Pauling's aggressive progress on DNA structure would make Wilkins feel urgent and seek cooperation with him and Crick. But even risking Pauling snatching the Nobel Prize, Wilkins and Franklin remained unenthusiastic. They hadn't even made any three-dimensional molecular models of DNA. Even when Watson offered Cambridge's model, they stayed unmoved. Time passed through Christmas 1952, with no new updates from Pauling. Watson guessed Pauling's model must have hit major trouble. Otherwise, he wouldn't stay so low-key. In February 1953, Pauling finally wrote his findings into a paper and submitted it to the proceedings of the National Academy of Sciences. When they opened the paper, everyone was stunned, because Pauling's model was identical to Watson and Crick's failed triple-chain nucleotide model from a year ago. Watson spotted a flaw in Pauling's model. Remember, DNA stands for deoxyribonucleic acid, but Pauling's model omitted the acid part. So Pauling hadn't created DNA at all, but DN. Watson and Crick couldn't grasp how Pauling, a world-renowned sharp chemist, made such an unforgivable error, but they at least breathed a sigh of relief. They still had a shot at surpassing him. Watson took Pauling's paper and visited Wilkins at King's College again. Wilkins showed him a new experimental report Franklin had just produced. The moment Watson saw the report's photo, he was dumbfounded. It was a much clearer X-ray diffraction image than any before. The DNA molecule showed a very distinct black cross pattern under the lens. Years of research experience told him only a helical structure could form such a stable shape.
At the report's end, Franklin concluded that the phosphate sugar backbone sits on the outside of the helix.
Watson acutely realized Franklin was incredibly close to the right answer. All she lacked now was verification through a molecular model.
If I wanted to unearth this treasure first, I'd have to hurry,
He and Crick plunged into complex chemical calculations involving phosphates, bases, and helices, quickly discovering DNA's two key structural features: base pairing and anti-parallel complementarity. Simply put, nucleotides have four bases: adenine, G, C, and thymine. That always equals T, G always equals C, meaning they pair up in molecular structures. And on the two nucleotide chains, they run anti-parallel, meaning if one DNA strand orders nucleotides 1 through 2 through 3 through 4, the complementary strand runs 4 through 3 through 2 through 1.
Thus, an unprecedented DNA molecular model was built.
They'd succeeded in building a double helix model with phosphate backbones, twisting externally and bases pairing internally, but they weren't fully confident it matched reality. The next scientific verification involved comparing this model's predicted diffraction pattern with actual X-ray data. Only if they matched could they prove DNA's double helix wasn't wishful thinking. To complete this, Watson and Crick had to turn to Wilkins again. Wilkins was astounded by their discovery. Despite these young men potentially claiming credit he might have deserved, he held no grudge and readily agreed. 2 days later, Wilkins called. X-ray diffraction photos powerfully confirmed the double helix's accuracy. Watson and Crick were thrilled but couldn't relax yet. In science, only published papers provide final proof. Collaborating with Wilkins meant King's College research team knew about their model. King's College was compiling these results into a paper to publish alongside the double helix article. Rumors suggested Pauling was revising his DNA model and even preparing to invite Watson to explain their discovery.
The race is now in its final sprint phase.
Watson and Crick worked through the weekend to finish their paper, delivering it to Sir Bragg, head of Cavendish Laboratory, on Tuesday. Now, Sir Bragg greatly praised their work. He wrote an enthusiastic recommendation letter, and by Wednesday, both documents were at Nature's editorial office. On April 25, 1953, Nature published Watson and Crick's paper, concluding one of science's most thrilling competitions. DNA's double helix discovery joins relativity and quantum mechanics as the 21st century's three greatest scientific breakthroughs. So what does it actually mean for humanity? DNA's discovery stands as life science's second milestone after Darwin's evolution theory. Darwin's breakthrough revealed connections between life forms, but not how they worked. The double helix showed this connection. Studying DNA's genetic code unlocks life's mysteries across all organisms. In near decades, technological advances from this discovery have deeply transformed our lives. As DNA research advanced, recombinant DNA technology became possible. We now modify genes to create pest-resistant crops and high-yield livestock, benefits that will keep growing. Gene editing may conquer deadly diseases and enable cloned organ replacements. Ultimately, Watson and Crick's contribution to scientific progress is immense. Some call them prodigies, rookies who outshone seasoned experts. But scientific progress isn't about lone geniuses appearing miraculously. Countless great minds stand behind every major achievement. While Watson and Crick come to mind first, other scientists, Nobel laureates like Pauling, Wilkins, and Perutz, made vital contributions to DNA's discovery. When we cheer for Watson's discovery of the DNA double helix, maybe we're not just praising how great he was, but also the dazzling era he lived in. Okay, this concludes our interpretation of the double helix, the story of the discovery of the structure of DNA. Reading notes are below the audio. We'll see you next time.